Okay, we're back. We're live. My God, it's Monday morning. Okay, it's Monday morning, and we are doing a show we've been trying to set up for a long time with an old friend, Cindy Goldstein. Cindy is the Industrial Relations Manager at DuPont Pioneer. And also we have Dr. Cliff Hudson. He's the Technology Lead Researcher at DuPont Pioneer. Wow.
No,
Am
just
I right
for double
about that?
haploids, just for one type of technology, just double haploidy.
Double haploid. Okay, that technology. Lead haploid technology.
Yeah, well,
Okay. And you're going to find out in this show about haploid technology. Anyway, welcome to the show, you guys. Cindy,
Great.
Cliff.
Aloha.
Nice to
Thank you,
meet you.
Jay. Hope you had a great Thanksgiving,
I did.
because food is important to all of us.
Food, you know, you can get along with it without for a few hours, but after that, it's just downright unpleasant without food.
That's right, and there are a lot of parts of the world where people aren't necessarily eating the kind of diet that we eat. And, you know, we oftentimes talk about some of the GMO or genetic engineering types of challenges and controversy. And, you know, it's easy when you have lots of choices at your grocery store, lots of choices for fresh produce that you can buy, to say, gee, you know, I don't like that technology and you shouldn't have it either.
That's the part. You shouldn't have it either. That really bothers me. It's also interesting that the whole issue about GMOs, it comes and then it goes, like a sine curve.
Yeah.
I remember it was all hot in, say, 2005, and then it went underground, nobody said a word, boo. And it gets hot again. It's sort of it's a generational thing. It's when there are no other protests going on. If you have no other protests, that's the
Yeah.
one.
That's the new
So
one.
what's the status of the controversy or the former controversy on GMOs now?
So I think, Jay, you know, you're a science guy, and for a long time we talked about the science and the safety. And it has been shown after many years of being in the marketplace since the 1990s that these crops are safe to grow, they're safe to consume, and I think many people, even that don't like the use of the technology, will admit that these crops are safe to eat and safe to grow. Now the segue we see is more of a philosophical viewpoint. I don't like those big companies. I don't trust those big companies and what they're doing with our seeds, and therefore they just don't like the technology being implemented, and
It's
especially
not
in
about
Europe.
the technology, it's about the big companies.
Yeah, and especially in Europe, and I think Cliff can speak to that too, the
Yeah.
extent to which we see that.
no.
Yeah, interesting. Well, what happened to the issue over pesticides? Is that gone?
Well, you know, that's interesting. That's where it's morphed to. So a
It's
lot
another
of,
morph.
yes.
When
it's-
you can't find traction on a given protest,
Yes.
you morph to another one.
Or you throw the spaghetti against the wall, you test which ones stick, and turns out the pesticide, raising concerns about use of pesticides. And I would like to take this opportunity with you to correct one big piece of misinformation. No plant requires a pesticide to grow. Genetically engineered crops do not require pesticides to grow. It's the choice of the farmer how they want to control the pests that they will encounter. And we support all types of farming at DuPont Pioneer, but do want to point out that organic crops do also, those farmers are using pesticides. They're approved for use for organic farming.
Well, if you want to do diversified agriculture, and we need, may I say it again, we need diversified agriculture in the state and elsewhere
Yes.
as a career and a source of food, instead of importing everything from Lima, Chile, Peru,
And other parts.
and
of
other
the
parts.
world that we can grow here, perhaps.
That we should grow here.
Yeah.
That's the point. And this controversy has actually hurt the small farmer. It's hurt diversified agriculture because they
Yeah.
get scared away. Nobody wants to, you know, it's really too bad. So, yeah, the other thing is, what about this notion that if you make a GMO in some, say corn, and then it dies automatically, and so the market requires another shipment the next year. So you perpetuate the demand. What about that?
So one of the things that we've seen over the years is misinformation or just deliberate fabrication of information. And, you know, a classic example of that was something called the Terminator gene. So how many plants out
Is that
there
Arnold
growing out
Schwarzenegger
there?
or what?
Yeah, well, yes, that was a good name they gave it, as opposed to, what was the previous term for it. It had some very scientific, flashy sounding name. But how many plants do you think that are growing out there in farmers' fields have the terminator gene? Think a bunch from all that you heard about it? About this many. 0. So it was a proof of concept in the lab, a way to control pollen flow and a way to, the mechanisms were used for various types of applications. And the number of plants that are growing out there in farmers' fields or growing anywhere with the terminator gene are 0. So for the amount of misinformation we heard about that, and purposeful misinformation, I think there are many people that were well aware that there were no terminator plants growing anywhere and still continued with that myth that the terminator gene is problematic.
Okay, one last question, and I want to go to the science, because
Yeah.
that's why we're here today. Who is making all this tumult? Who are the people who, create the nonprofits and fund the nonprofits that come and make all the protests? I mean, there's got to be a group, and I just don't know who they are or what motivates them, what
Yeah.
really motivates them.
Well, I think, it's broad what motivates individuals that get involved. There are a number of people that get fired up by social media, see a lot posted, see a lot that's worrisome. We talk about science, and we're here to talk about science and technology, and we're good at that. But we're not good at the emotions and about, some of the fear, frankly, that is put forward. And so for a long time, we talked about the science and the data. And really, all I need to say to you is, Jay, would you feed that to your keiki? What do you think about that? I don't know. We need to take a step back. All I need to do is raise doubt in your mind about whether it's safe to feed to your grandchild. And, all my science and data goes out the window.
Yeah, and that somehow intersects with this whole notion of you should eat well. But I think what I hear you saying, and I'd like you to confirm this for me, is that you can eat well, all those organic rules, all those natural rules, and still have GMOs, and you won't be injured.
Well, what's interesting, in the very early days of the National Organic Program from USDA for certification, it allowed GE crops because it was a way to reduce the reliance on a pesticide to control an insect. It was a way to use a technology for higher crop yields. And in the end, the public comment was that no, genetically engineered crops should not be part of that. But we support all types of agriculture. In fact, DuPont Pioneer is the largest supplier of soybean seed for organic farmers. So it's very compatible in our minds and can coexist quite well.
Yeah, so you can have a nice diet, a healthy diet, a diet you'd be happy to serve your grandchild with GMOs, because the world is living on GMOs anyway. We talk more about, the global demand and how that works later.
Yeah.
But for now, yeah, what else?
A lot of what we're doing in crop improvement is using some new tools. For instance, healthier foods. And I think we all want to eat healthier foods. And one of the things we've been hearing a lot is how we shouldn't have trans fats in our diet, how we should remove those or eat oils, eat a healthier soybean. So there's work that's been done to create a healthier soybean using RNA. It's called RNAi, or RNA interference, where you actually have multiple copies of small pieces of RNA which have reduced the amount of expression for one of the genes that makes an unhealthy fatty acid.
You're going into the science now.
I'm going
right?
into the
You
science.
Segue right into the science.
Yes.
You couldn't wait another minute. Okay, let's do that, okay? RNA, to my knowledge, came out only a few months ago, at least in the press. I think I read it in an MIT, tech circular. You can sign up for those. And it really dazzled me that this could be done, because this was not a GMO. It was an instant thing. It happened right now. You weren't making any permanent changes. You were merely controlling the expression of a gene at the surface level, at the expression level.
Yeah.
And that changes everything, doesn't it? That's new.
It does, because in the past, we would put in a gene to make something, a new protein. And now we're looking more at mechanisms that either turn off a gene or silence the gene, or we're looking at mechanisms that may reduce the amount of gene expression. And so, you know, when we're talking about RNAi, this is naturally occurring and was found, but can you tell me one genetically engineered crop that we grow here in Hawaii where that mechanism was involved in the resistance? Any idea? Papaya.
Oh, yes, papaya was an RNA science technology.
It is.
And Dennis Gonsalves at Cornell, right?
Yes.
He came back, he was determined to save papaya. It wasn't an option. If he hadn't saved papaya, there'd be a problem with papaya. Delicious papaya, healthy papaya, all the best things in the world papaya.
Indeed.
People will do somersaults on the mainland for papaya and in Asia, trust me.
Yeah,
So
yeah.
he saved it. He's a hero.
Yes.
Hey, Dennis, shout out to Dennis.
Yes,
Okay,
Dennis has done a great job. Came back here, worked with the U.S. Department of Agriculture over in Hilo, and continued to do excellent work where that turned out to be the mechanism. They introduced a gene for the resistance to papaya ring spot virus, which has saved the papaya here in Hawaii. And it turns out the mechanism for that was these small pieces of RNA, which then was the mechanism for giving the resistance.
Yeah, so what I read when I read this article, I think it was in the MIT newsletter, was that used to be when you wanted these small pieces of RNA, it was really expensive. But the processing to create these small pieces of RNA has gotten much more high tech. And now it's cheap. And you could buy enough RNA to affect a wheat field of several acres for $5. This changes really everything because it doesn't actually modify the genes. It modifies the expression of the genes temporarily.
Yeah.
So you have much more control. You sound like you want to say something.
No, I'm listening quite nicely here.
But
Cindy, what is your credentials?
I have a PhD in plant physiology and molecular biology from the University of Illinois, which means I was putting little pieces of DNA into plant cells and looking at their gene expression level. And then my master's degree is in plant breeding and genetics.
Okay. Cliff, how about you?
I was a biochemist. I have a bachelor's degree in biochemistry and ag biotechnology PhD from the University of London. And I'm also a fellow of the Royal Society of Biology in the United Kingdom.
Oh, that's great. And how long have you been working in this country?
25 years, actually.
All right, there we go.
Yeah. Started off in the Midwest and then came here in 2010. So,
Is Hawaii an important place for crop development? Yeah.
yes.
I mean, scientifically.
Scientifically, it's to be able to do the same sort of research that we do here, we would need huge amounts of growth chambers to have an environment that allows us to work year round. So the environment here is such a cooperative environment. Even when we have rainstorms, it still is a wonderful environment to grow all of our crops.
So one last question, we'll go to a break, but DuPont Pioneer here in Hawaii, it's a laboratory for DuPont Pioneer, but is DuPont Pioneer also creating food that I can eat here?
Yes, it comes back to us in many food products. So we primarily work with corn and with soybean, and we do some work with sunflower. And these products come back to us as corn oil, sunflower cooking oil, and so many things are made from corn and soybean that we might not realize. But yes, it
You're
just come
actually
back to
growing
us.
food here in Hawaii.
It comes back to us as food, but I would say what we're growing here in Hawaii is the seed,
The seed.
and then farmers will be
Yeah.
planting
Farmers
that
all
seed.
over the world.
Yes.
So a lot of the stuff that we have to do several generations. And because we can grow the material year round here, it allows us to do lots of generations that we wouldn't be able to do on the mainland. And that means that we can actually speed up our process to actually get to the end point. So the end point finally is material that's sold around the world, but most of the stuff here, it's the cycling that we do.
Yeah. And so you have an effect on the world.
Oh,
This is,
absolutely. This is really
a high-tech
crucial.
area for crop development.
Without
you know.
question.
Yeah.
Okay, we're gonna take a short break.
Okay.
We'll come back and we'll go further into the science. I know that's what you want to do.
Yes, very exciting.
We'll be right.
Aloha, my name is Justina Spiritu, and I'm the co-host of Hawaii Farmers Series. This is my co-host, Matthew Johnson, and you can catch us every Thursday at 4 p.m. at ThinkTechHawaii.com. What do we talk about, Matt?
So on Hawaii Farmers Series, we're going to be bringing on the farmers and also supporter of farmers, including restaurants, caterers, as well as government supporters and nonprofits to hear their background stories and understanding our local ag community just a little bit better.
Yeah, essentially, there's a lot more that goes into farming and the local food community beyond just producing the food. And we want to feature and get the background story on all these folks and see how we all work together as a community. So join us every Thursday. Aloha.
Okay, we're back, we're live. I'm Jay Fidel, and with me is Cliff Hudson on the far end of the table. He's the technology lead researcher on haploid technology for DuPont Pioneer. Okay, we're going to talk about that in a minute. And both of these guys are doctors. And Cindy Goldstein, she is the industry relations manager for DuPont Pioneer. And we are very happy to have you in the studio. You know, we can talk about the modern technologies. I don't think they get enough play in the press, honestly.
Mm-hmm.
No, and when it does, it's often portrayed in a controversial manner. You know, somewhere at the bottom of the article, it points out all the benefits and all the good things and the science behind it, and the front end is all about the controversy.
Yeah, right. And it must be something bad with it because all these people are saying there must be something, even if they don't know anything.
Mm-hmm.
Okay, so we talked a little about RNA. I consider that really interesting technology that can go huge places. And I'm sure that when the industry, when the researchers get their sleeves rolled up on this, there are going to be wonderful results with RNA technology. You know?
Yeah, a lot of new developments and a lot, this is a great time to be part of this field. There are so many new tools that can be used and so many new discoveries in genetics and how genes are regulated. It's a great time to be in this field.
Very exciting,
Yeah.
you know. Wish I'd gone back and taken some more science courses in college. I know I'd be with you on the other side of the table today, because this is so exciting. It not only crops, but any human thing. I mean, too. I mean, anything living, anything. So.
And there's a new technology called CRISPR, and I'm going to, that is
How do you
the
spell that?
Cluster.
CRISPR, the clustered, regularly interspaced, short palindromic repeats. I'm sure
It's gonna
I'll
be a short
know
test
that
at
name
the end of
soon.
the
Yeah,
show.
indeed.
But, and maybe Cliff would like to talk a little bit about CRISPR. It's a great new technology for plants, animals, all kinds of organisms.
Is this
Essentially-
newer than the RNA technique?
A little bit, but
Yeah.
I mean, it's been around. I mean, initially discovered in 1989, but then it's been in the last millennium, so 2002, 2005, and really it's become very popular now.
Well, how does it work?
Well, essentially it's gene editing. So it allows you to use a small piece of DNA to actually probe in, and they called it a guide RNA, and allows you to go into a particular location within the genome and surgically cut it. So you can actually have a very precise incision. So for a while, we weren't so good at just going for a single point, but with this technology, it allows us to do that. So you can
Little
either...
tiny scissors,
Ye-
really.
Absolutely.
Mhm.
So if there's
Yeah.
a single
Brilliant.
mutation, you
That's
can
it.
go in and correct it, or you can create one. So the thing is, you don't have to add anything. You can take stuff away, or you can just go in and switch things around.
How do you, I mean, methodologically, how do you do that? Do I have a very strong microscope and, no, how do you do that?
It's all enzymes and biochemical scissors.
Biochemical
we call
scissors.
them. Yes.
You heard it here on ThinkTech.
And I'll give a couple examples of this in the plant science world. So many people think about diseases as being, you might be susceptible to a disease, or a plant might have resistance, or a human to a disease. And so there's an example of a disease in wheat where there's one gene that gives the plant the susceptibility to the disease. And they just plain deleted the gene, and the wheat plants are now resistant to powdery mildew because they took away the gene that made it susceptible.
Now, you don't know if there are other effects, or you know that this gene has this effect, nothing else. And so when you do this, there's no third-party effect on it. It's just the effect you want. It's that precise.
So this hasn't been released. Okay, so before anything will be released, there will be much testing. So much as we would do with other crop modifications, we would actually have to have FDA approval before that will be released.
Yeah.
So your questions are really important, and we do need to pay attention to them.
Yeah. So, I mean, it's a matter of evaluating. I mean, of course, it's by surveys and databases and see what happens, kind of, sort of, I don't know what you call this in science, but, checking out the result on a database.
Yes.
But it's also about knowing exactly what that gene controls.
Well, and that's part of the application, is if you can knock out or delete a gene, then you can determine what is that gene doing. So if you're not quite sure if that gene is involved with resistance to a disease, or another example is with tomatoes. So one of the very early examples of a genetically engineered crop was the flavor saver tomato, which had delayed ripening. And I just saw a reference to the CRISPR technology being used, again with that same principle, where you can regulate the gene that slows the ripening of the tomato. So the CRISPR technology is going to be one of those paradigm shifts, like the 3D printers. It's going to be something that gives scientists an amazing tool that's going to be reliable and precise. And we even hear a lot about the use of this gene with the human genome and human embryos or animal embryos. It can be used to with many types of diseases.
Susceptibility to disease is the same idea, the same logic. It's, wow, the possibilities are enormous. Now, how does that differ from what you've been doing in the past in terms of trying to modify genes and DNA? How does it compare?
Well, the main thing is that you're not adding anything from a different species. It's already there.
So you're not splicing anything in?
You could, but for most of the applications that we're considering now, you would actually just be removing something or slightly modifying it from within the system itself. So no interspecies and activity at all.
Yeah, so it's actually simpler.
Yes.
Isn't
Oh,
it?
yes.
And I think what's going to be very interesting is how is this thought about in a regulatory sense. So we've not genetically engineered, we haven't inserted a new piece of DNA, and mutations are often just one change in one base pair or one base change that can be a mutation. And so it will not be possible to tell, was this a naturally occurring mutation or was this done using this CRISPR technology? And if you delete a gene, if you just plain inactivate it, well, then you haven't really done a genetic engineering step. And so I think it's going to be very interesting to follow the debate about this in the plant sciences, about do we call these transgenic or genetically engineered, because it really isn't inserting DNA.
Yeah.
And I think the other thing that's critical that we're going to see, I'm a big fan of Public Radio and Science Friday, so I was
So
listening
am I,
to...
by the way. Ira Flato was my hero. Yeah.
Hey, you're following his footsteps well there, Jay. We have to think long and hard about the technology, because it then will be passed on to future generations, that genetic change. And that's where especially, we're not, Cliff and I aren't so familiar with all of the human sort of applications and technologies, but just as you do reading, there was a New Yorker article that Cliff had shared with me that went into some of this. Very interesting to think about.
Very interesting, super interesting.
Yeah.
So the difference now between the RNA technique that we talked about first and the CRISPR technology that we're talking about now is that RNA affects the expression of an existing gene. You're not in there modifying the gene anyway. You're just modifying the expression on a temporary basis.
Right.
With CRISPR, you actually go in there and modify the gene.
Yeah, you use the RNA to actually find the gene that you're looking for. So you have that, it's got a very
exciting
specific,
stuff, yeah.
and then you use something called Cas9, which is a nuclease, which goes in and it just cuts in a very specific location. So you use the guide RNA to find out where you need to be, and then the cut. And you know, in the past, you had to introduce them at the same time, but now they're actually models in animals that you can actually, they actually have the Cas9 gene already present. So this is primarily for gene discovery. So understanding how genes are controlled and how they're expressed is one of the biggest tools, one of the biggest values of this technology.
So mapping the genome was only the first step.
Only the first step.
We're now into the detail
Yes,
of it.
absolutely.
We need to know about every
Yeah.
single gene,
Yeah.
what it does, and
Yeah.
if you take it out, what happens
Yeah.
then, so forth. Now, once you modify the gene, using CRISPR, for example, that's one cell, isn't that one cell? I mean, how do you make a field of new material based on this technique? How do you, deploy that?
Well, which brings me to my technology, which is haploid technology.
Okay, but I'm so excited about haploid that we need to take another break.
Okay.
Now, that happens to be Cliff Hudson. He's here with Cindy Goldstein. We're talking about, here on ThinkTech Talks, we're talking about crop technology at DuPont Pioneer. We'll be right back.
Aloha, I'm Kirsten Baumgart-Turner, and I host Sustainable Hawaii every Tuesday from 11 a.m. to 12 noon. My guests offer insights on challenging economic and environmental issues facing our state and offer innovative solutions to increasing Hawaii's long-term sustainability. Recently, we've been focusing on sustainable land development, food, and energy security. If you have a project or issue you'd like to discuss on the show or would like to be a guest, please contact me at kirstenbturner@gmail.com and tune in live weekly or view the show at your convenience at thinktechhawaii.com. Mahalo.
Aloha! How you doing? It's me, Angus McTech, asking you to come join us on ThinkTech Hawaii Hibachi Talk. Join me and my two hosts, Gordo the Tech Czar and Ender the Security Guy, every Friday from 12:45 till 13:45. See you on Fridays, and remember, let your wing gang free wherever you be.
Okay, we're back, we're live, and I want to be clear about this. It's Dr. Cliff Hunter,
Yes.
right?
Yes.
Okay. And Cindy Goldstein. So we were moving into this whole idea about haploid technology.
Yes.
And that's your specialty. So tell us how that works to get this special gene that's affected by the CRISPR technology out into a field or the world.
Well, luckily, we all start from a single cell. So either an egg cell or a sperm cell. And normally, in normal reproduction, they would fuse to produce a diploid individual. So what my technology does is we take either the egg cell or the sperm cell, and we create embryos from those, and then we diploidize them. So we double the chromosomes. So instead of having to have them fused, we actually allow them to develop, and then we form two sets of chromosomes so they're identical, because the first set of chromosomes is a template, and the second set is made, and you need to have two sets to make sure that the next generation is fertile.
We're talking about plants.
We're talking about plants, but you can do
Sperm
this
and egg
with
plants, yeah.
Absolutely.
Okay.
Pollen
Male and
grains.
female, pollen.
So
and-
pollen
Really?
grains,
Yes,
yeah,
yes.
indeed.
Pollen's male.
So,
Wow.
yeah,
The sex life of a plant.
absolutely, it's pretty
In
involved.
the next episode.
So the thing is, you can introduce or you can modify your gene in that single cell, and then you can diploidize that so that it's now capable of being fertile, and then you can create a whole organism from it. And the reason that we want to do this is that in our conventional breeding programs, it takes between 6 and 12 generations to actually get back to a parent line that we can use in a hybrid. And we need to have...
Mendelian.
Mendelian, yeah, absolutely.
Yeah.
And we need to have these fixed lines, so when we make a hybrid and sell it to the farmer, it always does the same thing. right? So if we go back through these single cells and we create plants from those, that allows us to do that in a single generation. And because we can do that in a single generation, it means that we can get to the end point more quickly. And
That's the export point.
well, the export point, yes, where we would actually have the fixed line that then we would send back to our breeders, and the breeders would test in their breeding programs for appropriate genetic composition.
How long does it take to get to the end point? So I give you now, one gene that's modified, and I say, Cliff, go for it. Give
Okay.
me an export, end of line, sell it to the world kind of
Oh,
product.
sell it to the world. Okay, so first of all, I have to sell it to the breeder. So first of all, I give it back to the breeder. So the breeder has to check that it does all of the things that he needs it to, or she needs it to do. Okay,
Who's the
so
breeder? Is the breeder
we
somebody
have.
with DuPont Pioneer?
Yes, so this will be within DuPont Pioneer, and these are scientists that they use all sorts of tools to actually understand the genetics of their individual inbred lines and how they combine with each other. So it would take us here in Hawaii about a year to return that material to them. They then test it. So when they test it, they test crossing with different parents in different combinations. And that could take 3 to 6 to 12 years, depending on the target and how difficult it is to do that. So if it's something else that it's like a GMO or something like that, which we don't work with here at the moment, would take extra time to be deregulated. Okay, so
When you say 10 to 12 years, you're including the, what do I call it,
All the
regulatory time?
Well, okay, if it's a GMO, yes. If it's just a regular, non-regulated material, then it could take 3 to 6 years to get enough data in enough locations to be confident that the product will perform exactly as we predict. So when we
is the
sell,
CRISPR.
yeah, well, okay. So the CRISPR we're still working on that. So we don't actually have any of that I'm aware
still
of
working
here
on
at the
it.
moment.
It's very
Yeah.
new.
So
When
this,
you say 10 years or
Yeah.
12, you're talking about Mendelian.
yeah, Mendelian stuff. Okay. Now with double haploids, you can cut 2 to 3 years off of that.
Okay.
So, and every time you get your fine product to the marketplace more quickly, you increase the MPV that you have for that. So it becomes more valuable to the company. So
Yeah, and for the world.
Yes, indeed. Well, we have to function at their grace. So,
Okay, let's move on to another, this is now, do you think this will occupy an important role in
Yes.
food going forward?
oh, absolutely. Understanding genes and gene expression and how we can use them to actually enhance the quality and the quantity of material is crucial.
Now, if I'm doing old-fashioned Hawaiian apu-a kind of agriculture, can I use your stuff?
Yes,
Yeah.
I can get your stuff and use it,
yes. So
and
it's-
I'll be safer. I can grow things in a safer way, more resistant to plant diseases and so forth.
Well, in fact, there are diseases that we cannot find any natural resistance. So with the papaya ring spot virus, which I had mentioned, they looked all around the world really at papaya collections to try to find resistance. And there were some plants that had small amounts of resistance, but really there was no resistance to be found just in the wild around the world.
This opens a very interesting question, because we live in a global village. And, where before, in early days, Hawaii was an isolated island, not too much came or went. It was, thousands of miles away from anywhere. Okay, now we have all kinds of influences coming in. Invasive species
Yeah.
at every level, not only, not only little things, but big things, and tiny things, and,
Yeah.
and all kinds of invasive species. So I may not, my local plant may not be able to survive because we can't find a natural resistance to a given invasive disease. So we must have this kind of technology
Yeah.
in order to save the plant, or the plant will be extinct. Am I right about this?
Yeah, I mean, these are tools that can be used when you know, we look at different things in plant breeding and often look for materials to work with. I'll give you another example of something like this. There is a disease called citrus greening, which is very
Thought you'd get to
problematic, which is very problematic in Florida, and we certainly don't want to have it here.
Yeah.
It's a bacteria that will impact citrus, mostly oranges, but also it can have an impact on grapefruit and on tangerine. And similarly, they've looked around the world for resistance to this disease. It happens to be tied to an insect that moves it, much like with the papaya ring spot virus, where there are insects that move the virus. There's an insect that moves this bacteria from plant to plant. And that is something where genetic engineering technology, where they took a gene from something called Arabidopsis, which is the lab rat of the plant world, tiny little kind of plant that is in the brassica family, in the same family as cabbages and
Canola.
cauliflower, canola, and mustard family. And by introducing a gene from the Arabidopsis plant, it then triggers a type of resistance mechanism in the orange plants, the trees that they've tried this with. And they have some plants that seem to be resistant to the citrus greening and some that seem to have better defense mechanism but not completely resistant. And so, you know, this is something terrible that we don't want to see spread to Hawaii or anywhere else, but real problem for the Florida citrus growers. Their yields expected for this year are much lower than what they've seen in something like 40 to 50 years.
Well, so what techniques are they using? What techniques do they hope to use to save their citrus? Save our citrus. I can see it now.
Well, some of it is to enhance the natural resistance mechanisms in the plant.
Using CRISPR.
This is using the standard genetic engineering of taking a gene from one plant, from Arabidopsis,
okay, as you said, you
putting
find
it
it in
in
one,
another plant.
And then you create the resistance in the other.
Yes.
And you therefore save the crop.
Yeah.
It would be a world problem to lose citrus.
Yeah, I mean, you say, okay, are these essential foods for us? Well, many people would say yes. It's a good source of vitamin C. Many people, you know, eat oranges and grapefruit as part of their healthy diet. Could the world live without oranges and papayas and grapefruit? Well, perhaps.
There's a lot of vitamin C tablets.
Well, yeah, and this is, you know, when we talk about an emphasis on healthy foods. So that mechanism is called systemic acquired resistance, SAR, where you introduce a gene through genetic engineering from another species that then triggers the enhancement of the response of the plant to the disease.
Important. And now this is in the research stage?
No,
It's
this
actually
is,
happening?
well, it's out in field tests. You know, much of this you do a lot of field tests to see if the resistance holds up over time. It's still, you know, a few years, maybe 3 to 5 years away from any farmer actually planting it.
Okay, let's talk about fish briefly, and then I want to talk about the global picture going forward.
Sure. So, you know, we started talking about, is there still controversy around this technology and around the crops? And yes, there is. And there's also controversy around the released GE fish. So there's a genetically engineered salmon that was created about 20 years ago. They took a gene from the Chinook salmon, took another gene from a creature that's a lot like a sea eel from the ocean, which keeps that gene turned on. So you have a salmon growth hormone, a gene for the growth hormone, and then you have something that keeps that gene active so that the fish grows more quickly than non-genetically engineered fish.
Is that like the RNA technology? It's an expression thing, or is it a change of the gene itself?
It is enhancing expression.
yeah,
It keeps it.
upregulates.
Genes are turned on and turned off by mechanisms, by different factors that will bind to receptors to turn on a gene or turn off a gene. There's something called methylation, which Yes, for another in-depth sort of blackboard
I
description.
love this stuff. This is great
It's
stuff.
fascinating how all this
It's
works.
really
And
the core of our universe, you know, our living universe.
And, you know, some people look at, you know, whether it's us that are part of seed companies or a company that's developing a new salmon, and they look at us like, I don't know about that. You know, when they developed the salmon, they made sure it is not fertile, so it's sterile, and they're growing it in controlled environments, not out in the ocean. So there's a company in Canada that is working with this technology. They're actually growing the fish, at least now the reports I've seen, in Panama in enclosed facilities. But there is concern that should these fish be in the wild, they would outcompete salmon that don't have the gene because they grow faster, they may be preferential for mating, and so there's a concern that if these were to escape into the environment, it would become the predominant type of salmon in the environment. So
Is that necessarily bad?
I think that's a
The world's salmon
philosophical,
supply is reducing all the time.
it's a philosophical discussion. I myself am not troubled by eating a genetically engineered fish or a genetically engineered corn or soybean, you know, tofu from a genetically engineered soybean. I have no
I think we all do,
concerns
actually,
about
at
that.
one point or another
Pretty
in our
much.
daily lives. You know.
But, you know, with the fish, now you're talking about the first animal that is in the market that is truly a genetically engineered animal.
Yeah.
And so that is what is novel. It took almost 20 years for this to be approved. In 2010, the FDA concluded that the fish was safe for people in the environment. December 2012, they did an environmental risk assessment, said it would pose little risk to the environment, and then it took until November of 2015 for FDA to make this approval.
Just happened.
It just happened.
A few days ago.
Yeah.
This
yeah,
month.
Yeah. So still very much, you know, in the controversial sphere of genetically engineered organisms. Part of it is the concept of it being in its environment, and then people having some concerns about, is there something about this that is different that would make it not healthy or safe for me to eat? And there's just no evidence for that.
Is it out there now? Can I buy this fish yet?
You cannot, but you can read numerous articles about all of the stores that we will not
That
sell
will
the
not sell.
salmon, which is not in the market. It'll be a couple years before we even see this in the marketplace.
But I think it probably will be popular and probably very healthy, actually. Salmon, someone said salmon is like the world's perfect fish. You know,
Yeah.
In terms of its nutrients and healthful aspects and its
Yeah,
taste.
yeah.
But let's go to the last thing. We only have really a minute left. And that is, why do we care about this? Why do we care? I mean, first of all, it sounds like we could not feed the world's population, which is increasing to over 7 billion, maybe 8 billion soon. At the same time, you know, our agricultural land is being taken for other uses here and elsewhere for development of one kind or another. And people aren't going into farming as much either. Those young people, even the ones who graduate from Sitar, they don't necessarily become farmers. So we need to use all the technology. But my question to you is, why do we care about these technologies? Why would an average citizen care?
It's our food. It was just Thanksgiving. We have so many cultural gatherings, and it's so much a part of our individual family heritage and family gatherings. Everybody now is tuned in to, or almost everybody, eating healthy, caring about what they eat, and it's very important to have safe and healthy food and an abundant food supply that's affordable.
Yeah, I mean, not enough food against having enough food. I think I know how to rule on that. We all have to eat. The growing number of people have to eat. And if this is what allows those people to eat, then we must pursue this line of technology. You want to close, Cliff, That's Cliff Hunter. I want to be clear
Yeah.
about that. Cliff, why don't you tell the people what you'd like to leave them with?
Well, I would like to say that the technology that we're using today helps us produce food better, but more to the point, I have a family myself, and I want to feel comfortable that I'm doing everything that I can to make sure that their life and the life of their children will be happy, healthy, and safe.
Thank you, Cliff.
My pleasure.
Cliff Hunter, a technology lead researcher on haploid technology in DuPont Pioneer, and Cindy Goldstein, industry relations manager from DuPont Pioneer. Thank you so much, both of you, for this great discussion.
Thank you, Jay.
Pleasure.
It was great to have a chance to talk with you about this.
Thank you very much for asking us to come.